So today's topic is transgenic mouse. So in the previous video we discussed some way that we can introduce a transgene which actually doesn't belong from the mouse genome, but we derived from another source. So in the previous video we have discussed some lentiviral vector method. So we will discuss this lentiviral vector method in detail in this video. So what we actually do, lentiviral vector in this method, we have some genome construct that I am just drawing it. This is called a 5 prime, which is a LTR. It is a 5 prime LTR, and this is a psi, which is unnecessary part, you can say. No, this is not an unnecessary part. It is different designation. It is a packaging signal and a PPT, which means polypurine tract, and another thing which is the most important thing, the transgene that we are trying to introduce through the lentivirus into the mouse. So at first we will integrate this transgene into the lentiviral genome, then the virus will infect the mouse embryo, then it will express the transgene in the mouse phenotype. So this is the overall concept, and after that there is a important thing, Woodchuck post transcriptional regulatory element WPRE and a 3 prime LTR. Okay, this is normal. So this is the actual genome construct, you can say, genome construct of the lentivirus, okay, which is lentivirus. So what we have done here, we just introduced this transgene in the, this is the whole lentiviral genome. So there is a 5 prime LTR, there is a psi, there is a polypurine tract, there is a transgene, and now you will think, what is this box? This box is the individual promoter for the expression of this gene, which is called a promoter, to express this gene in the lentiviral and further in the mouse. So what are the function of these specific elements to express this? So 5 prime LTR, there is a long terminal repeat, both present in 5 prime and 3 prime end. So it is help for first of all, you can write production of lentiviral RNAs. Okay, so this is lentiviral RNA production for 5 prime and 3 prime LTR. 3 prime and 5 prime LTR are needed for that. So next thing, this psi is actually a packaging signal. Okay, packaging signal, what it does, it helps to pack the lentiviral RNA, okay, lentiviral RNA into viral particles. Okay, particles. So my handwriting is a little bit bad in this case, so just remember this thing, this psi, this is a packaging signal, helps packages the RNA into the virus particle. Okay, so and the next thing is this actually the lentiviral is integrated into the host genome by the LTRs. We already know that this is the basics of virology. So this LTR helps to integrate to site specific recombine the lentiviral genome into the host, actually a DNA form, into a DNA form. And we have done with this thing, LTR psi. This is a polypurine tract. write down this polypurine tract, very important term, polypurine tract. So what it helps for, and actually if you consider PPT and WPRE, and I am writing this in a bunch. WPRE actually a woodchuck post transcriptional regulatory element. Okay, so you can, from the name you can predict that enhance the transduction of the host cell and increase the transgene expression in animal. This is the two term that enhance the transduction, these two things, remember this, enhance the transduction. I hope you know the basics of transduction. Transduction means the entry of that this lentiviral genome into the transgenic mouse host, not directly to the mouse but in the say individual cell. So to enhance the transduction rate, okay, so another thing is, and the enhance the transcription, directly you can say the transgene expression, it will increase the rate of transgene expression. Okay, and there is a additional detail which may not included, but I am including it, which is that there is a 3' in the 3' LTR there is a regulatory element is removed. This is a small detail. One regulatory element is deleted. Okay, so deleted for ease of transduction. But remember the important thing here, that transgene has its own promoter when it is inserted into the lentiviral genome. It doesn't depend on the viral promoter for the expression of that gene. So this is the whole thing of the lentiviral vector method. So if you have any doubts, write down in the comments. So thank you. See you in the next video.